Good morning, everyone. Welcome to this session. My name is Charlotte Teunissen. I will start sharing my screen so I can give you a proper introduction of this session. So I share my screen. So the title of this session is Accelerating the Development of Biomarkers to Identify Dementia. So it's an introduction to two European projects, the MIRIADE project and the bPride project. So first about the backgrounds, but these are my disclosures. We will give an introduction on the needs, why we need novel biomarkers, and then I give a very short introduction into MIRIADE and bPride. And then the other participants of this workshop, they will allude a little bit more on what these projects really entail. So they give some examples. So first, the need for biomarkers. We do need biomarkers to be able to make a biological diagnosis for Alzheimer's disease. We know that pathology starts already 20 years before we see the clinical symptoms. We know this for Alzheimer's disease, but it's likely also the case for other dementias. And we do need those biomarkers for the early diagnosis, because by definition, in these very early preclinical stages, we cannot rely on cognitive symptoms because they are not yet visible. We need those biomarkers for prognosis, so to define who will deteriorate quickly and who will not. And we need those biomarkers also for monitoring treatment effects, because by definition, we can still rely on clinical outcome measures in those very earlier stages. And biomarkers give us the opportunity to do an objective measure of the biological effect of treatments. And for Alzheimer's disease, we have very good biomarkers. So we can measure within the CSF or by PET imaging the levels of phospho-p-tau being elevated in CSF, and that's a reflection of the tangle formation. In addition, we can measure amyloid beta levels, which are decreased in the CSF, and these are a reflection of the plaques, because amyloid beta is the major component, the major protein inside the Alzheimer plaques. So we're very happy with that. So why do we need additional biomarkers? Well, it's not just amyloid and tau pathology. There are more mechanisms involved in Alzheimer's disease. And we want to understand the full complexity of the mechanisms, because that may also indicate which treatments we should apply and not just target only amyloid and only tau. So we need those biomarkers also for stratification to indicate, hopefully indicate who will react best to certain treatments. So here you see an overview of the biomarkers that we currently have, that we can measure already in the CSF, and others, and these markers, we can measure them nowadays also in blood. But this is focused on amyloid and p-tau, NFL and GFAP. So these markers, we know them, but we need additional biomarkers. There is another reason why we need additional biomarkers, and that is to improve the diagnosis of other dementias. So Alzheimer's is the major form of dementias, but it's not all. There are other dementias, and these also have difficulties, or it takes a long time before the diagnosis is made. So for example, in Lewy body dementia, it can take up to 4 years after the first visit to the clinical doctor to make a final diagnosis. So we need biomarkers to really discriminate those dementias as well. But there are challenges in biomarker discoveries. And to understand that, I want to explain to you how the biomarker development process usually goes. So it first starts with biomarker identification, and you have to come up with a biomarker. And we do so, for example, by screening by a technology that we call mass spectrometry proteomics. And then in the second phase, phase 2, we do some initial validation, and there we want to apply different technologies, usually immunoassays, because immunoassays are the assays that we use in clinical routine. So these are the ones that we can use for large-scale implementation ultimately. In the second phase, we apply those novel assays and we perform large clinical studies, for example, longitudinal studies. And then in the fourth phase, we perform prospective studies So we've done all the well-established clinical cohort studies, but then we are going to test the novel biomarkers in the real world. And in the last phase, we prepare everything for implementation. So let me draw your attention to this first step. You see immediately that these are two different technologies. And we think that that's probably one of the problems. So we do have a challenge. So in the past, we had difficulties to translate our mass spectrometry results to those immunoassays. So what could be the reason for that? So we thought of several options. One was that we compared our CSF, so in the cerebrospinal fluid, based results obtained by mass spectrometry proteomics with those in post-mortem results. So same technology, but now in people that died of Alzheimer's disease. But that did not solve our problem. We still had a lot of failures. Another issue is a larger N, so more patients in your discovery studies, because we typically had included only 10 per disease group, 10 patients per disease group. So that's not so big. And there were other reasons that we don't know yet. But one of the reasons that we thought would be very relevant is that we made a switch. So we, in the technology, we started with mass spectrometry in our discovery results, and in the validations, we used a different technology and different types of measurements, the immunoassays. So we thought that that could be one of the problems, the causes of our failures. So, but now we thought of a solution. There are novel methods nowadays available that allow us to do the discovery not by mass spectrometry, but by immunoassays. So we don't have this transfer in technology along our experiments. And that's the basis of the MIRIADE Marie Curie project, and also of the bPride project. And the method that we use for that is the O-link, that's the company, proximity ligation assay. Now, that's a technical detail, but that's the assay that we use. So within the MIRIADE, we want to tackle all our problems. Sorry. We do so by training 15 PhD students to innovate a biomarker development for dementias. So we want to cover this whole process from biomarker identification to implementation. We call it here bring to the markets. And we have an extra layer of process innovation. And how this works with assay development, from biomarker to assay development, biomarker identification, sorry, to assay development will be explained by Katharina Bolsewig in one of the next presentations. And Leighann, Kimble, and also Ekaterina, Mavrina, they will explain how we want to address the process innovation and how that's tackled within MIRIADE, because that's an overall work package and a task within MIRIADE that we want to address. Finally, we will show some data, a project that's designed to develop blood biomarkers as main focus. Because we can measure now these CSF biomarkers very well for Alzheimer's disease, but we want to have blood-based biomarkers. That's much more convenient. And for that, we designed the bPride project. So again, we will apply the O-link technology to identify novel biomarkers. And again, we are addressing AD, DLB and FTD for optimal discrimination of each of these diseases. And that will be presented by Marta del Campo. So this is my introduction, and now I give the floor to Katharina Bolsewig. And here are my acknowledgements. It's relevant.
So yeah, hello everybody. My name is Katharina Bolsewig. I'm a second-year PhD student at the Amsterdam UMC, and I'm working in the group of Charlotte Teunissen as part of the MIRIADE project. And I'm very happy to tell you a bit about my project in particular today, which is about biomarker development for dementia with Lewy bodies. So when we're talking about dementia, we're actually using an umbrella term which describes a wide range of symptoms that are all associated with cognitive impairment. However, there are different causes for dementia, such as Alzheimer's disease, which is the most common form, but also less frequent forms, such as dementia with Lewy bodies, which I'm going to focus on here. But in general, we already see an overlap when it comes to symptoms and also pathology between these different groups, which makes the differentiation of these often a challenging task. And if we're talking about dementia with Lewy bodies, we're talking about a Lewy body disease. So in Lewy body diseases, we see the accumulation of different proteins into so-called Lewy bodies inside of the brain. And depending on where these Lewy bodies accumulate, which area of the brain, we can differentiate the Lewy body diseases even more. So one type is dementia with Lewy bodies, as I just said, but also Parkinson's disease, dementia, and Parkinson's disease is one type of Lewy body disease. And if we're looking even closer into the dementia with Lewy body group, we see that some subjects have a pure DLB pathology, whereas most of them actually have a mixed pathology with Alzheimer's disease. So this indicates again that there is a big symptomatic and pathological overlap between diseases, and this unfortunately can lead to misdiagnosis, which again can lead to mistreatment that might even worsen symptoms, and in general we can observe a long diagnostic process. And the diagnostic process includes several tests and examination. Not only is the individual medical history of each patient considered, but also physical and neurological examinations are being performed, mental and cognitive tests, brain scanning, such as the DAT scans, and the protein measurements of the cerebrospinal fluid biomarkers for Alzheimer's disease to exclude Alzheimer's disease as a diagnosis. And some of these tests are being perceived as an invasive procedure for some of the patients, but also some of them, especially the brain scans, can be very pricey. And on average, we see a 2 years time span until a final diagnosis is being made, which of course is a bit too long, or we prefer it to be shorter, which is where our work comes into play. Because if we would have a disease-specific biofluid biomarker, we could possibly accelerate this process, and we can also make it less pricey and more convenient for the patient. And if we're talking about biofluid biomarkers, what do we actually mean? So a biomarker in general is a measurable indicator of some biological state. And in our case, we want to measure proteins in different biofluids that reflect the disease state and help us to differentiate different types of dementias from each other. And when we're talking about the biofluids here, we are focusing on 2 biofluids, cerebrospinal fluid, or short CSF, and blood. And both of these have their own advantages and disadvantages. So cerebrospinal fluid is the fluid that surrounds our brain, and due to the direct contact with the brain, it is thought to most precisely reflect the ongoing processes in the brain and give us a very brain-specific picture. However, we will need a lumbar puncture to sample the CSF, which can be perceived as rather invasive for some patients, which is why ideally we would like to have a more accessible biofluid, for example, blood, which is very easily being sampled and can be even drawn at the GP. In blood, however, we need to consider that the concentrations of the proteins often are way lower than what we measure in the CSF, so we need a way more sensitive assay to measure these, and they can also be affected by more processes in the rest of the body. So for us, when we approach the biomarker assay development, we first focus on CSF to make sure that we have a very specific biomarker, and then at a later stage, we like to translate this assay into blood so we have a more accessible biomarker. And for the rest of this presentation, I will now focus on the CSF. So as Charlotte already mentioned, for biomarker development, we go through these different phases. So we first need to identify an interesting protein that could be a biomarker for our intended use. We need to develop and optimize an assay for the detection of this protein. We need to then clinically validate that we actually can really differentiate between groups using this protein. And in the end, we need to make it our assay market ready so we can implement it also in the clinic. But this whole process can be very lengthy and can take up to 20 years, which is why we thought that, as Charlotte already mentioned, if we are making use of the same principle, namely using antibody-based methods for both the biomarker identification step and the assay development step, we could maybe shorten this time span. So what we did was first to set up a big proteomic study where we sent our samples to the Swedish company Olink, and they are making use of the proximity extension assay technology to detect different proteins in the CSF by using antibodies. So this is quite technical, but I In short, they are using antibodies that are detecting thousands of proteins in our samples, from which we can then generate a signal, which is being translated into a normalized concentration value. And from these results, we can then analyze which proteins might be more interesting for our intended use. And for these ones, we can then go into the immunoassay development step. So here you can see the setup that we're using in our immunoassays. It's called the so-called sandwich setup, where we are using a primary antibody that is capturing our protein of interest, and a secondary antibody that detects the same protein and is bound to an enzyme that can, upon addition of a substrate, create a colorimetric or fluorescent reaction, which we can then translate into a concentration. And so as you see, in both of these steps, we're using antibodies to detect the proteins. Only in the first step, we're detecting thousands of proteins in one sample, and in our immunoassay, we only want to detect this one protein. And now I would like to show you some results we got from our proteomics study, because for dementia with Lewy bodies, we were able to identify a very interesting biomarker candidate. This is the protein dopa decarboxylase, or short DDC. And if you look to the left in our volcano plot, you see a lot of proteins. And very distinguishedly, you can see that DDC is an outlier here. And what this graph tells us is that when we compare control subjects to subjects with dementia with Lewy bodies, DDC is significantly increased in the dementia with Lewy bodies group. And also when we look to the right, to the box plots, we see that this increase is not only observed when comparing dementia with Lewy bodies to controls, but also when we compare it to Alzheimer's disease. So we really see that we can, at least from these preliminary data, differentiate these groups from each other, which of course, especially for AD and DLB is very important because of the shared pathologies. But not only from these very striking proteomics results, DDC seems promising, but also if we look at the function itself. In general, DDC decarboxylates aromatic amino acids to the corresponding amines. And in particular interesting here is the decarboxylation of L-dopa to dopamine, which we already know is a key player or is affected in alpha-synucleinopathies. So we know also that DDC is a rate-limiting step in dopamine synthesis in the Parkinson's disease. And in cell culture experiment, it has been shown that there is an interaction between alpha-synuclein and DDC. And in alpha-synuclein overexpressing cells, a decreased activity of DDC was observed. So all of these results together really made it seem that there is possibly a very important role of DDC in dementia with Lewy bodies. So we decided to continue with developing an assay to detect DDC in CSF. And we first approached it by making use of an enzyme-linked immunosorbent assay, or short ELISA. This is a manual assay which makes use of the same sandwich setup that I just showed you. And we tested here 4 different antibodies and their combinations with each other. And for these antibodies, we tested different concentrations and also different buffer conditions. However, we always encountered a sensitivity issue of the ELISA here. So that means we were not able to generate a signal for DDC in CSF, and we were not able to measure the DDC with this assay. So we then continued the assay development using a more sensitive platform, the Simpleplex ELA platform. This is the principle of the Simpleplex ELA. So it is an automated ELISA, meaning, again, it makes use of the sandwich setup. However, now less hands-on time is needed. And the reaction is taking place in such a cartridge that you can see on the left-hand side, in which we have a microfluidic channel system that you can see on the right-hand side. And in these microchannels, we see 3 glass nanoreactors for each samples, in which actually the sandwich setup is located. So that means where the protein is being detected. And so for each sample, we are measuring triplicates here. And when we translated our assay from the ELISA to the ELA, we were now able to detect the DDC in our CSF values. And when we're looking at the signal to noise ratio, we actually saw an almost 7-fold increase of our signal to noise ratio in the CSF sample. And in order to make sure that our assay can also accurately determine the CSF concentration, we then performed a technical validation of the assay, of which I would like to discuss the results for the parameters sensitivity, precision, parallelism, and dilution linearity with you now. So when it comes to sensitivity, we want to make sure that the lowest concentration that can be accurately measured is below the concentration that we measure in our samples. So the lowest limit of quantification that we determined was 5.24 picograms per milliliter, which falls here between the third and fourth lowest calibrator of our calibration curve. And the measured concentration of DDC in the CSF samples on average lays at around 30 picograms per milliliter, which falls higher in our curve, meaning we can accurately determine our concentration. Next, we were looking at precision of our assay, and here we were actually looking at two things. The first parameter being the repeatability or intra-plate variation, which gives us an indication about how much our measurements vary between same samples of one run. And in general, we accept a variation of 20%. So what we see for repeatability is that we can very nicely accept this parameter because it only has a coefficient of variation of 8.6%. The next parameter would be intermediate precision or interplate variation that tells us how much our measurements vary between different runs. And here we were measuring an intermediate precision of 21.5%, which is a little bit higher than the accepted value. However, due to technical complications, these two measurements were only based on 2 separate runs, which is why we are very optimistic that with the further measurements, we will be able to decrease these values even more. Next, we're looking at parallelism. And here we would like to know if the antibodies we're using for the detection and capturing of our antibodies are behaving in the same way to our protein inside our CSF samples as they do to the calibration curve where we are using recombinant protein. And the accepted range here would be 85% to 115%. And very nicely, again, our measured values fall within this range. And I think you can also very nicely see this in the graph, where not only the samples are parallel to each other upon dilution, but also are parallel to the calibrator that is depicted here in green. And lastly, we were looking at dilution linearity, again, accepting the same values as for parallelism. And again, our measured values fell within that range. And we were able also to see that even upon very high concentrations of our sample, we would not observe a hook effect, which is very nice. So to summarize it all, we were able to identify a very interesting biomarker candidate, namely DDC, as a potential biomarker for dementia with Lewy bodies. We were able to develop an assay that is able to detect DDC in CSF on the ELA platform, and we proceeded with the technical validation. And in the next steps, we would like to assess the sample stability of DDC and would like to perform a clinical validation to see whether or not we can actually differentiate different clinical groups by making use of the DDC measurements. And we would like to also develop an even more sensitive assay on a different platform, for example, the CIMOA platform, where we might even be able to see smaller changes of DDC concentrations. So that could also possibly be very interesting if we are looking if we're trying to differentiate different Lewy body diseases from each other. And with that, I would like to thank you all for your attention. And I would like to thank Alzheimer Europe for giving me the opportunity to speak here. And I also like to thank all my colleagues from the Amsterdam UMC and also from MIRIADE, of course.
Thank you very much, Katarina. So I think at the end, we hopefully have some time for discussion and questions. I don't see questions yet in the Q&A. So please, attendees, if you have questions, you can ask them in the Q&A. And now we continue with the presentation. It's a duo presentation of Ekaterina Mavrina and Leighann Kimble. So I give you the floor.
Thank you very much. So my name is Leighann Kimble, and I'm joined by Katharina Mavrina, and we are each second-year PhDs, and we'll be excited to talk to you about the innovation process in biomarker discovery. So our work is a little bit different in the sense that we're utilizing qualitative research methods, and I'll explain a bit the benefits of that and the opportunities that are allotted for understanding this process. As you've seen with Kat's presentation, this process is quite complex, and biomarker discovery occurs over a long period of time. And so by looking at process innovation, which I'll explain a bit more, we have the opportunity to really understand how this process unfolds and hopefully be able to use this information to improve research advancements and innovation in the future. And so given we've already seen this image a few times now, and what's important about this image is that MIRIADE provides us with an interdisciplinary context. And so along the way, in terms of biomarker identification, assay development, what we're really focusing on here is the process innovation aspect of this. And that requires data sharing and reuse as well as collaboration. And so within this whole process, we're looking at different actors, we're looking at different fields, disciplines, and of course, the dispersion geographically across the different actors and different organizations that are involved in this initiative. And so what we're looking at is innovation as not this linear or closed process, which we see here. So the idea is that you start with an entry point and then you end up with a product or research advancement, but instead looking at innovation as an emergent or open process in which we have a lot of different practices that are occurring that we need to follow, and it's not quite so easy to understand. And so what we really focus on is how can we synthesize the dynamics in which innovation is occurring as a process. And we emphasize here the open aspect because this concept of data sharing, reuse, collaboration between partners and organizations is really the key to allowing these research advancements to move forward. And a lot of times we don't have the opportunity to take a step back and understand how this process unfolds. And that's the role of Katya and myself in MIRIADE and understanding what this looks like. And so in order to do this, what we are using is MIRIADE as essentially a living lab, because we don't have a lot of individuals who are taking that step back and looking at what's occurring during biomarker discovery. We are taking this opportunity to use qualitative data collection approaches to understand what is happening in MIRIADE. What does innovation look like? What are the processes that are occurring? And that means for myself, it will be focusing on the data sharing and reuse aspect. And that goes hand in hand with Katya's work, which she'll explain is this concept of collaboration and coordination. And we do that by, rather than using the equipment and looking at the measures that are taking more so on the quantitative side and in the labs, we use interviews, observations, documents, and site visits to understand what our researchers are doing and really capture their processes along the way. And so for the data sharing and reuse piece, I really highlight in my own research the understanding of openness within MIRIADE. And so my question is, how does data sharing and reuse occur within a collaborative context? And what I highlight here is the importance of novel discoveries. So what we see in MIRIADE is not that we're reusing data for reproducibility. So we're not essentially seeing if we can reproduce the same results. And what that means is that we're reusing data in a very different way for novel discoveries. And that requires data reuse in a way that is not using the same data to see if the results are occurring exactly the same way, but rather reusing this data in a way that is not standardized. So we don't have an approach for novel discoveries. And so because it's beyond this concept of reproducibility, it comes with unique challenges and of course associated with solutions as we go along this process. And all of these occur in context. So it's both the context of the project, the actors, the organizations, but also the data itself. So recognizing that data is produced in context and therefore that's going to have an impact on the way it is able to be reused. And so returning to the idea of openness, as I've highlighted, collaboration, data sharing is central to allowing us to make novel discoveries. So all of the work that's being done by MIRIADE builds upon the work that was prior to others, it builds upon the literature, it is informed. And so that requires having openness in terms of data, science, and innovation. But as you can see by this very complex image, it looks quite different for each of these areas. So if we look at, for instance, open data, we have the idea of data sharing. We're advancing research science by sharing this data and reusing the data. With open science, we're really focusing in on research. So having collaboration in MIRIADE, it's also involving patients in addition to the different research groups and disciplines, and in our case, research approaches. And when we see open innovation, we look at more so our industry partners in terms of how can they utilize this coordination and sharing, collaboration to advance their own project goals and advance innovation. And so within this, what I'm also looking at in terms of the data sharing and reuse is what this means for openness in each of these different areas. And what are the limitations when we think about openness between data, science, and innovation? Because that does play a role in how we're able to translate our research into innovation in the end. And so hopefully that means exploring this image a little bit more and tying it to empirics. And the overall goal is having this idea of openness for doing better science, and that's central to my research. And I think also the heart of Myriade. And so transitioning over to the work of Katya, data sharing and reuse means that there's collaboration, there's coordination that is ongoing, and that requires both openness but governance. We need some sort of structure for collaboration, and that occurs in practice. We have exchanges between actors. We can't do this individually. We have to work together and share knowledge, data, resources in this practice. And then, of course, returning to the idea of situated context at these various levels, ranging from the individual to context in terms of geography as we are dispersed. And with that, I'll turn over to Katya, who will be speaking with you a bit on the collaboration and coordination.
Yes, thank you, Leighann. So in my research, I'm focusing on how collaboration and coordination unfold over time in interdisciplinary consortia, and why it's important to see how collaboration and coordination change over time. I'll explain in the coming minutes. So first of all, given that biomarker discovery, assay development, and commercialization are various steps. They require the input from various research teams. And for example, biomarker identification is done in Myriade by computational scientists such as data scientists. We also have bioinformaticians. But assay development, clinical validation is conducted by biochemists, neuroscientists. So these are very different research teams that have different backgrounds, different approaches to solving problems, and they use different methods for achieving the goals within their own projects. And although collaboration is a desirable thing, but given that these are very diverse research teams, they face difficulties in understanding the needs of each other and in anticipating where they could be helpful for each other. That's why it's important for us to understand what bottlenecks and what problems the teams face when trying to interact with each other, and also to see which teams tend to interact more intensively and understand the reasons for this, and which teams prefer not to. That's why I'm saying that we focus on collaboration dynamics, so seeing how collaboration emerges and evolves over time. On the other hand, what we have in Myriade is that, well, there is actually no firm, there is no university that possesses all necessary competencies, knowledge, resources, tools, equipment that it could use for doing all the stages in this innovative process. And it requires joint effort of various organizations. And you can see here that in Myriade we have a lot of universities, firms, and institutions involved. And they are spread across the globe. So as a result, coordination and orchestration of processes becomes also challenging, because it's completely different from having the teams located in one organization. Here we have various institutions, and they are autonomous, they are independent, but they share a common goal to advance research in the area of dementia. So that's why coordination is also something that is evolving over time, and it's affecting collaboration, but also collaboration can affect coordination mechanism used in such big interdisciplinary consortia as MIRIADE. Next slide, please. So as I am saying, so first, my first study deals with understanding how collaboration could advance complex and uncertain process of biomarker development by looking at coordination and collaboration as they emerge and evolve over time. And specifically, we are interested in understanding what difficulties the interdisciplinary teams face, but also how they overcome the tensions in their processes, misunderstandings, and how they can be helpful to each other so that they can, in the end, go through all those steps from biomarker identification to commercialization of assays. Next slide, please. Yes, thank you. And the second question that I also aim to address in my research is to understand how academic institutions, so it means universities, and how industry, which means corporations, firms, could collaborate efficiently when they try to solve such complex and uncertain innovative problems as research in dementia. Because, well, universities and firms, they have usually a little bit different focus in research and in their practices, because academia is more on the side of basic and exploratory research, doing a lot of experiments, while industry is usually associated with research closer to the market. And that's a benefit because we can bring the results which are done within universities to the market so that it can go and help to patients. But on the other hand, the divergence comes from approaches, for example, of academia to publishing. So publishing quickly, making results public, while industry firms may prefer to keep some of the findings in secrecy and might opt for intellectual property protection. So obviously there are some similarities in goals, but there are also differences in approaches in targets. And so as a result, we wonder to see how these two types of organizations, namely academia and industry, could really help each other overcome the tensions and the differences in their processes, goals, and practices, and how they could maximize complementarity so that they could really advance research in the area of dementia. Next, please. And so we, as the researchers in the area of social sciences, business, and innovation management, we also seek the input of the audience from the Alzheimer Europe conference to encourage discussion and to hear your stories on the 4 topics, because we are collecting data, we still conduct interviews, so it will be of great help for us if there are people in the conference who have the stories to share with us on, first, data sharing, reuse, and integration. Second topic, collaboration experiences in interdisciplinary settings. The third topic is the role, insights, and interests of individuals in research. And the fourth one, we also would like to hear from experts in immunoassays and mass spectrometry and other methods. And how can you contact us in case you have the stories to share with us? Well, very easily, our emails. And we would like to thank you very much for this opportunity to share our research interests. And we hope that we will be able to contribute to our understanding of how such big consortia, which really need a joint effort of various interdisciplinary groups, how they could operate efficiently to make advancements for solving societally vital issues as dementia research. Thank you very much.
Yes, thank you very much. And now I give the floor to Marta del Campo. It's nice. Yeah, sorry, Marta, you were the second to speak.
Yeah, I noticed, but okay, never mind.
Yeah.
No problem, I think. There's no problem.
But now we continue with the hot topic of blood-based biomarkers. So yeah, Marta, please go ahead.
Well, so good morning, everyone. I'm very glad I can present here our data from a project that we recently got funding from the European Union Joint Program of Neurodegenerative Diseases Research. And through these presentations, I will try you to explain what brings us to this project and the first steps that we have done. So as it has been highlighted before, dementia is a disorder that is characterized by a progressive impairment of cognitive function. It affects reasoning, emotional control, and memory. One of the major contributors or risk factor affecting to the development of dementia is aging. And due to increased aging population, it is expected that actually by 2050, more than 130 million of people will be suffering from this disease worldwide. And actually, it has been calculated that almost every 3 seconds there is another person diagnosed with dementia. So of course, this would create a huge impact for patients and the families, and also for the social and economical system. So we need to solve this, and in order to create new treatments, and we need also novel diagnostic tools. And as Charlotte has very well explained before, one of the tools that are very widely used are biomarkers. So we really need to have biomarkers. So it has been already mentioned that dementia is actually an umbrella term that covers different types of dementia. So if we zoom in into the brain of patients suffering from dementia, we know that most of the patients are Alzheimer's disease, which are characterized by the aggregation of amyloid proteins and phosphorylated tau. But there are also other types of dementia. We have dementia with Lewy bodies, which are characterized by other types of aggregates called alpha-synuclein, or frontotemporal dementia, which are actually even more complex because we can find 2 different types of protein aggregates. But it's all about these protein aggregates. Is everything? Are these protein aggregates causing the dementia? The truth is that what Charlotte also already mentioned is that new research pinpoint that there is not only these hallmarks, these protein aggregates that cause dementia, but there are many other types of biological processes involved in the development of the dementia. So, for example, we know that there are problems in the immune function or in the vascular function, or also in those connections that neurons make in order to create memories, on the energy. So there are many processes that might be involved in the development of this dementia. And therefore, we really need biomarkers that allow us to depict this all biological complexity along the different types of dementia. These biomarkers can be used for early and specific diagnosis of the different dementia types, might be useful to predict who will develop dementia or not, to monitor in which stage each patient it is, or even to see whether a treatment that is currently in a clinical trial is actually being effective or not, whether it modulates these different processes or not. And of course, it's also important to keep in mind that the earlier we can detect them, those changes, the better, because then we will be able to tackle the disease before the completely loss of neurodegeneration has taken place. So where can we find biomarkers? Well, there are different sources from the fluid, from the body, where we can find biomarkers, as it has been already shown. The main one that has been widely used is cerebrospinal fluid, because it's in direct contact with the brain. And more recently, we are really getting great advancements in blood, which of course is much more easy to get and to work with. And what can we detect in these body fluids? Well, we can detect different types of things, different types of components. One are the proteins, such as the proteins that we see in the brains of these patients, but we can find also different types of metabolites of DNA and RNA. In our group, we mainly focus on the detection of these proteins. And I guess that by now you are very familiar already with this workflow. This is the workflow that we have thoroughly used to develop novel biomarkers. we start with a first phase of identification. As Charlotte mentioned, we usually in this phase we use mass spectrometry approaches. What happened with these approaches? That in this approach we really have to cut all the proteins in pieces, and then we detect these different pieces to see which is increased and which ones are decreased. This is a very laborious method, and therefore it only allows us to use small sample of patients. Once that we know which proteins we are interested in, we then develop an immunoassay that allows us to directly detect the proteins of interest. Since we do not need to do such a laborious process, we can really now analyze these proteins in a much larger sample size, allowing to really validate the findings, and also these assays are supposed to, we can use it later on in the clinic. But what we have noticed in all our work is that we barely managed to go through this stage to the end of the workflow, and we observed that it was highly challenged to translate the findings that we observe here, to translate it in immunoassays that for large screening. But luckily, in the last year, there has been a huge advancement from a technological perspective, and there are new technologies that we think may help to fill that gap. These technologies, what allows now is to directly measure the proteins without any pre-processing. So therefore, it's much more simple and allow us also to analyze a large sample of patients already since the discovery phase. Moreover, we can then directly use the same reagents to detect the proteins that we are interested in, and again replicate it in a different sample set. The good thing of these technologies is that they are optimal to analyze proteins both in blood and CSF. It can analyze as many samples and many proteins at the same time. All these proteins cover different biological processes, so all the processes that might be involved in the development of different dementias. And a very important asset is that we can actually use the same reagents that we use here can be used later to this phase. Of course, everything has the pros and cons, and this is, of course, has also some things that are not so good. And of course, we have to acknowledge that we cannot measure with these technologies all the human proteins. Something that might be able to do with mass spec is more freedom. There are more freedom with those technologies. But still, we can analyze more than 1,000 proteins, which is quite a lot. So therefore, we think that this is an optimal technology to develop novel biofluid biomarkers, as we expect that by using the same reagents, as I already said, we can probably bridge the gap and smooth the translation between discovery findings and further validation. So the first step that we did to test that was then we create the bPride study. In the bPride study, what we aim is to identify proteins that are able to discriminate dementias, and we did that in the CSF. So we analyzed a large number of CSF samples from the Amsterdam dementia cohort, some healthy controls, as well as patients that are in the first stages of Alzheimer's disease, so in very early stages, patients with Alzheimer's disease, and a group of non-AD dementia. So we took all these CSF samples and analyzed the protein content using these innovative technologies. And we divided the project in two parts. In the first part, we will check which of the proteins allow us to discriminate the different groups, and in the second part, we will try to use the reagents of those proteins to develop an assay that allow us to validate it later on in other cohorts. So here, some of the first results. Each line here represents a protein, and the higher it is the line, the higher the levels of the proteins. And here we have the different groups. The control group, the patients with early stage of Alzheimer's disease, patients with Alzheimer's disease, and patients with non-Alzheimer's disease. So what do we see? We observe that there are many proteins that they have normal basal levels. So they have basal levels, but as soon as the disease develops, the levels of these proteins start to increase. So they are increased already in very early stage of Alzheimer's disease and Alzheimer's disease. However, when we check those proteins in those patients that doesn't suffer from Alzheimer's disease but have other types of dementia, such as dementia with Lewy bodies or frontotemporal dementia, we observe that those proteins are not increased. So these proteins, if we look at them, what they do, we observe that they cover many of the different mechanisms that are known to play a role in Alzheimer's disease. So basically, this is a proof of concept that with this technology, we can actually detect specific 80 biomarkers in the CSF that reflect the diverse biology of Alzheimer's disease. So then we move to our second step. Can we translate these findings into assays with the proteins of interest? So here is just an example of one protein. And here you have the protein levels, the levels of that protein in our discovery cohort in the previous data that I showed you. So it goes from 0 up to 14. And here with the new assay that we have developed, and it goes from 0 to 14. And here each dot represents a patient. So we can actually observe that the patient that saw the lowest levels of this protein in the discovery phase, in the previous image that I showed you, showed also the lowest levels with this new assay. And if you continue with that, we also see that the patient with the highest level in the previous phase show again the highest levels in this with this new assay, indicating actually that the levels strongly correlate with our new developed assay. So actually, it seems that we can overcome that translation gap and the translation challenge that we face in our previous studies. So we were very excited about this data. We know that our approach works in CSF, but of course, this technology also allows you to measure the proteins in blood. So of course, blood is nice to work with because, as I said, it's easy. It creates less burden for the patient, and it also allows you to then get several aliquots along the disease process. So you can do a repetitive screening and see how the different biomarkers change along the disease process. So then, with this in mind, we build the bPride project, which is the same as the Pride project, but in blood. And therefore, we aim to identify blood proteins that allow us to discriminate the different dementia types. To do so, we have screened a large number of samples from patients covering this dementia. We employ these innovative technologies, and we hope that we can really detect the specific changes that will allow us to discriminate the different types of dementia. So how can we do this? How are we able to perform this project? A major asset and important thing that allow us to perform this project is the faithful engagement of the patients providing their samples for the analysis. Actually, if you can see here in this image, the disease has different phases. Here is the first phases, the medium phases, and in the late stage of the disease. And you can see that this corresponds a little bit with the clinical syndrome. So we have a preclinical phase in which pathology is already taking place, and a prodromal phase, and then and a dementia stage. And actually, we got samples from Alzheimer's patients covering all this workflow, all these different stages, and not only for Alzheimer's disease, but also for samples from dementia with Lewy bodies and frontotemporal dementia, and which is very important, and I think it's something quite unique from this study, is that we managed to have samples from patients that were follow up later on, and we know that with time they develop Alzheimer's dementia with Lewy bodies or frontotemporal dementia. So we actually even have samples from prodromal cases of other dementia types. And together, as I said, we have more than 1,000 samples. So how have we able to perform such an ambitious project? Well, we were able to do so thanks to a great collaborations across different specialized memory units across Europe, which are depicted here, and also our great collaborator also in Australia. It's also important to note that these different centers store always the samples in the same conditions, so we can really be sure that the changes that we identify are only caused due to a disease process. So how far are we? What have we done in this project? So this is the first part of the project. What we have done is we have collected the samples, the blood samples from each partner and each center. These samples covers different dementia types at different stages, as well as non-demented control. We have analyzed all the proteins on these samples using these innovative technologies, and we use also we analyze also the classical proteins that are known to be involved in this disorder, such as amyloid beta or p-tau. What we are currently doing is to check whether we can find those specific proteins that discriminate the different types of dementia. So we are really in the first phase of the discovery. And then what we plan to do is, once that we know which proteins are important for each disease and each disease stage, we will proceed to the development of the assay, as we do it already for the CSF project. And in the latest stage, once that we have the assay, we will validate it again in a large cohort of patients. So we hope that by the end of the project, we will have a diagnostic test that with a simple blood draw allow us to discriminate the different groups of patients. So I would like to finish just to highlighting that this is a highly collaborative project, that it can only be done joining forces across different centers worldwide. So thank you very much for your attention. And
yeah, thank you very much, Marta. I think it's very clear. And also thank you, Ekaterina, and Leighann. So we are in time, and there's time for some questions. Are there questions from the audience? Maybe you can, I don't know how it works, if you can raise hands or ask your questions in the Q&A. So for Marta, those assays that you're developing, which technologies do you use for that? Are these technologies that we can use in various laboratories, and are they easy to implement?
Yeah, thank you. So we are using different approaches. One is the one that Katharina showed. We have other people working with other markers also. But what we have done is to use exactly the same technology to develop such as the PIA, the Olink based technology. We are developing into multiplex panels similar to ELISA, and we hope that works. It looks that it is working. And whether this is possible to implement in the labs, well, it would be needed, of course, to buy a specific technology to measure that. But in theory, yes.
Okay, let's hope.
Yeah.
And I have a question for Leighann and Ekaterina, maybe first Ekaterina. Now you've worked about a year with the different ESRs. Is there something in the collaboration? The ESRs are the early stage researchers in the MIRIADE project. Is there something that you can already share from your experience that's what you found remarkable?
Yes, of course. Well, I would say that the researchers that are involved in assay development and validation, they really need to do a lot of experiments, and they face a lot of uncertainty because they do not know whether the experiment's gonna work or not. So it's a lengthy and time-consuming and complex process that they really need to focus on and to put a lot of effort into that. And so it requires the time for these teams to obtain some results that they can later share and communicate to other colleagues, to other teams. But for computational scientists like bioinformaticians or data scientists that we have, they have a slightly different approach, because I guess they also do a lot of exploratory work, and they create their models and they learn from data. but I guess that it's a little bit faster. They can obtain results a little bit faster. And so that's the reason why there is like the difference in terms of speed of achieving results and different type of uncertainty involved, because the experiments are different. And yeah, the nature of work is different. And of course, it's well known that communication between scientists that represent very like different disciplines, it can be difficult, and they really need to put effort into understanding how they could help each other, so that yeah.
Can you already give tips how people can understand each other better?
I think, yeah, I think it's important so that early stage researchers do not hesitate to contact, to get in touch with each other, and maybe to hear, even if you do not anticipate that maybe computational work, it seems like not very helpful for you, but probably in a conversation, you will discover that there is value. And I heard from some ESRs, and actually Katharina was sharing it with me, that she was envisaging value, for example, of bioinformatics for her own work. And that's great that we have this in MIRIADE.
Yeah, so that's the sharing expertise. So Katharina, do you want to add something to that or share your example?
Yeah, so I think it's a really nice consortium that we have there, and it's so nice to have these multidisciplinary work that we're doing. And especially when it comes to, for example, prediction of antibody binding sites that one of our ESRs is working on. This is really interesting for us when we have some more information on antibodies. So for myself, so far, I did not have enough information yet, but actually what I just heard the other day in the lab, that one of our colleagues not working on MIRIADE is actually contacting now one of us from MIRIADE, and they are working together. So it's also really nice that not only within MIRIADE we have this collaborative spirit, but we can also spread it to the rest of our groups.
So the disciplines are really mixing and benefiting
definitely
from each other. That's nice, nice. And Leighann, do you have some first observations that you can share?
Yes, so I think also extending on Katya's point, one thing that we see in terms of the idea of openness of data is just, if you make it more open, it's going to be reusable. But when we're looking at novel research, that's quite difficult. And I think it also goes with this collaboration point. You're not necessarily sure of what data or resources or knowledge that you have that's going to be useful for others, or that even understanding what all it is that you may need to advance your own research. And so it's that idea of openness. It also requires putting everything out there, whether or not it is useful, which seems sort of counterintuitive to being helpful to others. But I think because the consortium has so many resources and so much data available, a lot of times it is just pushing that exchange so that it's made available to everyone, whether or not it may be perceived as useful.
Oh, that's a good point. And do you think that, oh, I see a question from the Q&A, but I will, we still have time. Yeah, so may it also depend on the stage of in the career of the researchers, because the ESR, they all just started and they're exploring their projects themselves. So maybe you cannot really estimate yet what the value is of your findings and your experience for others. And maybe often people think, as you said, that it's not valuable for others, but it can at the same time be very valuable for others. Yeah.
Yes, I think that's exactly it. I think the ESRs have the double challenge of they're starting off, all of us are starting off in our careers, and also starting off for the other ESRs in brand new research. And so it's not as though, you know, you're reproducing a study and you know exactly what to look for based on the information of others. And so I think it's also having that exchange, feeling comfortable with those that may be a little bit more advanced in their career, and having those direct contacts with others in other organizations that may be also able to support that, because that's where the knowledge piece comes in, and also recognizing how to engage in knowledge seeking for the data and resources that may be needed as they go along in their research.
So I see a question from the Q&A, and it's not completely specified, but I think it's for Marta, maybe nice to answer. Do you envisage these markers being applied in clinical healthcare settings in the future, and how easy would this be to do?
Yeah, that's a very good question. I think is the holy grail. The truth is that we haven't been able to do so yet with any of the previous markers. We are very close to do that, I would say, now with the blood-based biomarkers of amyloid beta and p-tau and GFIP. So still, we acknowledge that we will have several challenges, but it's possible. I think we are closer than we have ever been. Now, with these new markers, it will take time. We will see for the clinic for sure in the GP, let's say, we will need to translate them to blood. That's why mainly we designed the bPride also. And hopefully, yes, I think that we have learned really a lot. So, I mean, let's say that we have been 20 years to develop these amyloid beta, p-tau markers from CSF to blood. We had a lot of challenges there. We have learned a lot, and we will apply those learnings now, and it will smooth the process. I also hope just that the fact that it's blood will make it easier, because it's easier to get more samples and to have more people characterized.
Maybe someone else wants to address it. So yeah, maybe Leighann, how do you foresee that this will help healthcare in the future? Your studies and the sharing.
I think overall, one of the opportunities that Katya and I have is to understand what are the challenges overall, as well as using that information to advance further research. So hopefully being able to capture the challenges, whether it be within Myriada, but we're also inviting insights from outside of Myriada to help support the Myriada project itself, but also looking at how can we further advance, hopefully more rapidly, research, because that is translating into innovation, which means, you know, bringing about social change, supporting patients, etc. And that requires actually understanding the process first.
Yes, and I think it's important to stress here that's the overall goal of Myriade. We have different instruments, and we alluded on a few here, how we want, but the ultimate goal is really to accelerate biomarker development. So we hope that we are able to accelerate this lengthy process of biomarker development for that has been for 20 years or even longer for the Alzheimer's CSF biomarker. So we want to do it quicker. And we think that this is an important proof of concept, how we can improve this workflow that can be translated to other neurological diseases, but also to other diseases in general. Because yeah, we think that this is that's our a priori hypothesis, that this way it will help us in developing biomarkers for any disease, fluid biomarkers. So I want to thank again all the speakers for their valuable contributions. I think that you gave a good outline of what Myriade is and also what bPride is and the concepts that we're working with. And I thank you for your nice discussion, and also thank the audience for their participation and being there. So enjoy your day.